Good day, this is Professor Will, CBMD PhD. Today is Tuesday, June 23rd, 2020. It is 3:21:41 seconds a.m. Eastern Standard Time. And this is a continuation of my Corona series. But before we get started here, I wrote a little message of the day. So let's get started here. A little something I wrote. Today's message of the day. As you can see, I'm digging deeper into research and discovering and exploring a whole lot more. I'm going places where I've never gone before. And I could talk at great length of the things I recognize throughout my observations and scientific scrutiny. The scientific tools of today are ancient artifacts. I need to reinvent more accurate diagnostic scientific instruments for the laboratory. That being more distinct diagnostic tools to be able to explore where no man has gone before. I enjoy inventing, discovering, improvising, and working in my own workshop, especially on scientific instruments. Hopefully my discoveries, inventions, and time will make a big difference in the world. As a philanthropist, my dream aspirations and goals are for world peace. Critically ill COVID-19 patients are 10 times more likely to develop cardiac arrhythmias. Studies suggest cardiac arrest and arrhythmias are likely triggered by systemic illness, not solely due to the viral infection itself. Date June 22, 2020. Source University of Pennsylvania, School of Medicine. Summary. Patients with COVID-19 who were admitted to an intensive care unit were 10 times more likely than other hospitalized COVID-19 patients to suffer cardiac arrest or heart rhythm disorders, according to a new study. Researchers say the results suggest that cardiac arrest and arrhythmias suffered by some patients with COVID-19 are likely triggered by a severe systemic form of the disease and are not the sole consequence of the viral infection. The findings, which differ significantly from early reports that showed a high incidence of arrhythmias among all COVID-19 patients, provide more clarity about the role of the novel coronavirus, SARS-CoV-2, and the disease it causes, COVID-19, in the development of arrhythmias including irregular heart rate, atrial fibrillation, slow heart rhythms, RAD arrhythmias, or rapid heart rate that stops by itself within 30 seconds, non-sustained ventricular tachycardia. In order to best protect and treat patients who develop COVID-19, it is critical for us to improve our understanding of how the disease affects various organs and pathways within our body, including our heart rhythm abnormality. Our findings suggest that non-cardiac causes such as systemic infection, inflammation, and illness are likely to contribute more to the occurrence of cardiac arrest and arrhythmias and damage or infected heart cells due to the viral infection. Recent studies from China have suggested that COVID-19 is associated with a high incidence of cardiac arrhythmias, particularly among critically ill patients. Early reports showed 44% of patients admitted to the ICU suffered arrhythmias. Heart rhythm problems occur when electrical impulses that coordinate your heartbeats don't work properly, causing your heart to beat too fast, too slow, or irregularly. If left untreated, cardiac arrhythmias can lead to serious medical conditions, including stroke and cardiac arrest, the abrupt loss of heart function. To evaluate the risk and incidence of cardiac arrest and arrhythmias among hospitalized patients with COVID-19, the Penn team evaluated 700 patients with COVID-19 who were admitted to the hospital at the University of Pennsylvania between March and mid-May. Researchers evaluated cardiac telemetry and clinical records for patients' demographics and medical comorbidities, such as heart disease, diabetes, and chronic kidney disease, and recorded patient vitals, test results, and treatment. The cohort of patients had a mean of 50 years, with black patients accounting for more than 70% of the population. Researchers identified a total of 53 arrhythmic events, 9 patients who suffered cardiac arrest, 25 patients with atrial fibrillation who required treatment, 9 patients with clinically significant bradyarrhythmias, and 10 non-sustained ventricular tachycardia events. The team did not identify any case of heart block, sustained ventricular tachycardia, or ventricular fibrillation. Of the 700 patients hospitalized, about 11% were admitted to ICU. None of the other hospitalized patients suffered a cardiac arrest. After controlling for underlying demographic and clinical factors, researchers found cardiac arrest and arrhythmias were more likely to occur among patients in an ICU compared to other hospitalized patients. Researchers noted that the study has several limitations, including that the analysis was conducted from a single center serving a large urban population. More research is needed to assess whether the presence of cardiac arrhythmias has long-term health effects on patients who were hospitalized for COVID-19. In the meantime, it is important that we launch studies to evaluate the most effective and safest strategies for long-term anticoagulation and rhythm management in this population. COVID-19 lockdown reveals human impact on wildlife. Date June 22, 2020. Source Max Planck, Gesellschaft. Summary. An international team of scientists is investigating how animals are responding to reduced levels of human activity during the COVID-19 pandemic. Many countries around the world went into lockdown to control the spread of COVID-19, brought about by the most tragic circumstances. This period of unusually reduced human mobility, which the article's authors coined anthropods, can provide invaluable insights into human-wildlife interaction. There have been countless posts on social media over the past few months, reporting unusual wildlife encounters. Anecdotal observations, especially from metropolitan areas, suggest that nature has responded to lockdown. There not only seem to be more animals than usual, but there are also some surprising visitors. Pumas have been spotted prowling the streets of downtown San Diego, Chile, and dolphins recently showed up in untypically calm waters in the harbor of Trieste, Italy. For other species, the pandemic may have created new challenges. For example, some urban-dwelling animals, like gulls, rats, and monkeys, may struggle to make ends meet without access to human food. In more remote areas, reduced human presence may potentially put endangered species, such as rhinos or raptors, at increased risk of poaching or persecution. The authors emphasize that society's priority must be to tackle the immense human tragedy and hardship caused by COVID-19. But they argue that we cannot afford to miss the opportunity to chart, for the first time on a truly global scale, the extent to which modern humans' mobility affects wildlife. To address this challenge, researchers recently formed the COVID-19 Biologging Initiative. The international consortium will investigate animals' movements, behavior, and stress level before, during, and after COVID-19 lockdown, using data collected with nifty animal-attached electronic devices called biologgers. All over the world, field biologists have fitted animals with miniature tracking devices. These biologgers provide a gold mine of information on animal movement and behavior, which we can now tap to improve our understanding of human-wildlife interactions, with benefits for all. The team will integrate results from a wide variety of animals, including fish, birds, and mammals, in an attempt to build a global picture of lockdown effects. So what do scientists hope to learn? Researchers explain that it will be possible to address previously intractable questions. We will be able to investigate if the movements of animals in modern landscapes are predominantly affected by built structures, or by the presence of humans. That is a big deal. These insights will in turn inspire innovative proposals for improving human-wildlife coexistence. Nobody is asking for humans to stay in permanent lockdown, but we may discover that relatively minor changes to our lifestyles and transport networks could potentially have significant benefits for both ecosystems and humans. Coordinated global wildlife research during this period of crisis will provide unforeseen opportunities for humans to forge a mutually beneficial coexistence with other species, and to rediscover how important a healthy environment is for our own well-being. Viruses can steal our genetic code to create new human virus genes. Date June 18, 2020. Source: Mount Sinai School of Medicine. Summary. A new study unveils a novel mechanism that allows viruses to produce unexpected proteins. Like a scene out of The Invasion of the Body Snatchers, a virus infects a host and converts it into a factory of making more copies of itself. Now researchers have shown that a large group of viruses, including the influenza viruses and other serious pathogens, steal genetic signals from their hosts to expand their own genomes. The cross-disciplinary team of virologists looked at a large group of viruses known as the segmented negative strand RNA virus, which include widespread and serious pathogens of humans, domesticated animals, and plants, including the influenza viruses and Lassa virus, the cause of Lassa fever. They showed that by stealing genetic signals from their host, viruses could produce a wealthy of previously undetected protein. The researchers labeled them as UFO, upstream Frankenstein open reading frame proteins, as they are encoded by stitching together the host and viral sequencing. There was no knowledge of the existence of these kinds of proteins prior to this study. These UFO proteins can alter the course of viral infection and could be exploited for vaccine purposes. The capacity of a pathogen to overcome host barriers and establish infection is based on the expression of pathogen-derived protein. To understand how a pathogen antagonizes the host and establishes an infection, we need to have a clear understanding of what proteins a pathogen encodes, how they function, and the manner in which they contribute to virulence. Viruses cannot build their own proteins, so they need to feed suitable instructions to the machinery that builds proteins in their host cell. Viruses are known to do this through a process called cap snatching, in which they cut the end from one of the cell's own protein encoding messages, a messenger RNA or mRNA, and then extend that sequence with a copy of one of their own genes. This This gives a hybrid message to be read. For decades we thought that by the time the body encounters a signal to start translating that message into protein, a start codon, it is reading a message provided to it solely by the virus. Our work shows that the host sequence is not silent. The researchers show that because they make hybrids of host mRNAs with their own gene, viruses, SNSVs, can produce messages with extra host-derived start codons, a process called start snatching. This makes it possible to translate previously unsuspected proteins from the hybrid host virus sequences. They further show that these novel genes are expressed by influenza viruses, and potentially a vast number of other viruses. The product of these hybrid genes can be visible to the immune system, and they can modulate virulence. Further studies are needed to understand this new class of proteins, and what the implications are of their pervasive expression by many of the RNA viruses that cause epidemics and pandemics. Researchers say viruses take over their host at a molecular level, and this work identifies a new way in which some viruses can wring every last bit of potential out of the molecular machinery they are exploiting. While the work done here focuses on influenza viruses, it implies that a huge number of viral species can make previously unsuspected genes. Researchers say the next part of their work is to understand the distinct roles the unsuspected genes play. Now we know they exist, we can study them and use the knowledge to help disease eradication. A large global effort is required to stop viral epidemics and pandemics, and these new insights may lead to identifying novel ways to stop infection. Good day, this is Professor Will, CBMD PhD, on infectious disease, molecular microbiology, and pathophysiology. We hope you enjoyed listening. Until next time, have a great day.